So we've heard wonderful things already about man's best friend and how much they are affiliated with us, as demonstrated by their brain activity. Then we've heard from our rabbit whisperer and his excellent sharing of information about what he's been able to do with housing rabbits in groups. We're now going to switch gears and hear from our fish whisperer, Doreen Bartlett, who is currently at the Office of Laboratory Animal Welfare at NIH, but spent many years with managing the fish facilities at the National Institute of Child Health and Human Development. And then we will be hearing from Jim Cox, who is our frog whisperer, and he is at the Genelia Farm facility of the Howard Hughes Medical Institute. And then lastly, we'll hear from Eric Hutchinson, who says he's not a pig whisperer, but he does know what a pig looks like. So that's who we have up next. So to start with, Doreen.
Good afternoon. Good morning, isn't it? We haven't had lunch yet. First, I'd like to appreciate the opportunity to speak at the symposium, and I commend the incorporation of aquatics into this symposium. So I've been working with OLAW for the last 3.5 years, and my talk today is not to be considered OLAW directive, but I do want to speak about the requirements of the guide as they apply specifically to zebrafish, and to share what some institutions out there are doing in the way of socialization and enrichment, and as well as some of the very interesting research to date. So I hope you'll come away from this talk with appreciation of the various ways that environmental enrichment is being and can be addressed with zebrafish, with the goal of improving the overall health of the zebrafish and its value as a research animal model. So many times we get blank looks when asking about provisions of environmental enrichment for zebrafish, not to mention the rolling of the eyes. This actually is a very interesting area, and there are so many variables involved. The only wrong answer is that it's not needed for zebrafish. You'd be surprised on how it's already being incorporated into zebrafish husbandry without people even realizing what they're doing. Okay, so just to touch on some of the NIH guide requirements that specifically to highlight use of zebrafish. The primary aim of environmental enrichment is to enhance animal well-being, and this can be done through structures and resources, which facilitate the expression of species-typical behaviors. And just like all other environmental factors, it should be considered an independent variable and therefore appropriately controlled. So a lot of researchers, surprise, working with zebrafish, have raised concerns that environmental enrichment may compromise their research. But we also need to keep in mind, if we're working towards the goal of providing an environment where they can exhibit species-typical behavior, this can improve welfare, reduce stress, and hopefully reduce animal use. With that said, a fish is not a mouse, is not a monkey. It's very important to recognize the many non-structural options available when using zebrafish. We're not trying to make it into something it's not. So let's look at the guide as it discusses aquatics. Environmental enrichment strategies for many aquatic species are not well established. What is the impact of barren versus enriched environments? And when used, enrichment should elicit species-specific appropriate behaviors and be evaluated for safety and utility. Generally, schooling or shoaling fish species are housed with conspecifics. And aggression in zebrafish does occur. Therefore, monitoring and intervention is important. Improved breeding success has been seen in enriched environments, but of course, further research in this area is needed. Nowhere does the guide say that you must or even should put a physical object in every tank. So the areas of the guide on the left outlining the aquatic microenvironment considerations also should be considered with any form of environmental enrichment. The size of the tank is associated with normal physiological and behavioral needs, as well as allowing conspecific social interactions such as shoaling. You shouldn't disrupt the foods in the vertical water column. It needs to be determined if and how an in-tank enrichment will be cleaned. Can it even be autoclaved? It shouldn't pose a problem with filtration by blocking the overflow or breaking apart. It shouldn't cause physical harm with sharp edges, entanglement, entrapment of appendages, or unintentional ingestion, and it should restrict escape. Zebrafish are very good jumpers. It should be constructed of non-toxic materials that do not leach toxicants or chemicals into the aquatic environment. This is very important. And it shouldn't present electrical hazards, directly or indirectly. It should permit the ability to observe the fish with minimal disturbance, and the nature of the research should be taken into consideration. Zebrafish strains are known to differ in their behavior and propensity for stress and anxiety, while there is no difference in terms of behavior and aggression between the sexes. So an example here of the Easter grass, there was a very proactive researcher many years ago at one institution that the people came in and found Easter grass in the zebrafish tanks. And while she was very well-meaning, the grass could have entrapped the fish, it could have clogged the overflow, the fish could have ingested it, and most of all, it could have been toxic. So there's a fry test that's widely accepted, and this is a very sensitive test, as the larval stage is the most sensitive stage. It clearly shows the effects that otherwise would not be noticed, but it only tells the immediate toxicity and not what may be breaking down or leaching into the system over time, affecting gene expression as well as morphological and physiological changes. And just a reminder that you would need an IACUC-approved protocol for this procedure because you're using zebrafish over 3 days post-fertilization. Okay, so what is normal? Let me see if I get this right. So this is a fish and distribution in the water column, shoaling of fish. It's allowing conspecific social interactions. They're very fluid, no pun intended, but this is normal behavior. Then, in recognition of stress, this is in typical housing tanks for zebrafish. The fish in the large tank on the left are displaying the normal shoaling behavior, while the fish in the large tank on the right are displaying the classic stress response. This tank on the right had just been put on the system, which had a vibrating pump, and the fish were not used to this. So when first exposed to a novel environment, the zebrafish have a tendency to linger at the bottom and sides of the tanks. This is compared to Thigmotaxis and rodents. And they have a prolonged hesitancy to enter the upper half of the tank. And they'll do this after a while, but it will take a little while. They also have increased anxiety-like behaviors such as erratic movements and freezing bouts. Then we look at the impact of social interactions with stocking density. So this is the impact of a higher stocking density, about approximately 15 fish per liter. This is about 30 fish in the tank. The lab animal article, Enrichment for Laboratory Zebrafish, stated that little evidence to suggest that zebrafish maintained at adequate housing densities require additional enrichment devices. Also, the Zebrafish Journal, with the article Husbandry, a Zebrafish Danio and cortisol response, indicated that when group sizes increased to greater than 10 fish, social hierarchies and the group were reduced and the group displayed a normal shoaling behavior and lower cortisol concentrations. However, with that said, schools of fish need space to express their physiological and behavioral needs, and their exposure to inappropriate water volume of less than 2 liters, even in a good water chemistry, results in increased stress. Then we look at an intermediate stocking density of approximately 8 fish per liter. And you can see some of these fish now are darting around in the water. It's not that real fluid motion. The enrichment for laboratory zebrafish article indicated that aggressive interactions between zebrafish are most frequent at low holding densities, which may be manifested in reduced reproduction, growth, and immune function. Then at the low stocking density, we have 2 fish in a tank, and you see the constant aggression that typically occurs when 2 zebrafish are kept in the same housing tank. And this is negative for both of the fish. So animals whose well-being is compromised by being placed in unsuitable social groupings are often physiologically and immunologically compromised and therefore have an adverse impact on the quality of the scientific data. So here, an artificial plant has been added to the low stocking density, and these are 2 adult zebrafish in a 1.8 liter tank with the subordinate fish using the plastic plant as refuge from the dominant fish, showing the aggressive behavior. So one institution has a provision of supplying artificial plants to ensure that aggression is minimized in any fish maintained under the density of 5 fish per liter. Another study showed that a preference and behavioral response to increased structural complexity, where zebrafish spent twice as long in a compartment equipped with artificial plants and clay pots as compared to a barren compartment. Also, when a laboratory tank was split into 16 areas, of which 7 contained artificial plants, zebrafish could be found in those 7 squares 99% of the time. Now, what's interesting about this last bullet point and the laboratory tank split into 16 areas is that they were both at lower stocking densities of approximately 8 fish per liter. So it'd be interesting to see what would happen if you did the same testing with your 15 fish per liter. So why would zebrafish be singly housed? They are aggressive, so animal welfare reasons, or for the identification of the fish themselves for scientific reasons. Right now, there are no microchips available for zebrafish that are small enough. I did read an article. There are some RFI chips that have been used on zebrafish, but I have not seen that used in the industry yet. When you test the zebrafish individually housed, they're more stressed, they have more variable behaviors due to disruption of their natural shoaling strategies, and another study had suggests that adding an artificial plant as enrichment to the single-house zebrafish when you couldn't socially house them. Another interesting one was an institution, if you could tell a difference in the zebrafish phenotypes, that maybe one had the long flowing tail and the other ones had the spots, that you could possibly put the populations together. So then we see also in breeding, we definitely see the aggression in breeding tanks. And when you overlook the factors such as substrate and the presence of the natural plant material, we're limiting their natural behavior and expression, and in doing so, we may be negatively affecting their lay rates. Also, if you have fish that have been housed singly for, say, identification purposes, you really need to, or welfare reasons, because you know they're aggressive, you really need to be mindful when you put them together in your crosses that there may be possible aggression shown. So one case study was done where they had tanks with no vegetation, with vegetation, and a controlled tank of individually housed males. The difference between the no vegetation and vegetation, it didn't demonstrate a reduction in cortisol associated with providing the vegetation, but it did prevent injury and death from the fighting. So this is especially true whenever you have single pairs of zebrafish that must be maintained for more than 24 hours. Interesting, though, the control group, the individual males, there were no deaths, but they had the largest increase in cortisol over the other 2 groups. So then we're adding the artificial plants to breeding tanks. So zebrafish are known to prefer to spawn in sites associated with aquatic vegetation. This is also an example of the depth gradient. find the right button. Okay. The depth gradient, where you have your tank on a tilt like this, so that it provides a shallow spawn area for the fish. And also at this time, I'd like to thank Chris Lawrence of Harvard Children's Hospital in Boston for the use of his wonderful videos in this presentation. And if you want to see any other videos of Chris's, just go to YouTube, Google Chris Lawrence, and he has some amazing videos. Okay, there's another case study. This was done at University of Texas Medical Center, where they put plants in chambers to encourage breeding. And they were looking, well, do they like grass plants? Do they like leaf plants? Do they have a preference for plants? And they actually found that, number one, of course, they liked the plants. It provided them sanctuary against their aggressive partners. And I believe this is going to be published in JLAS. I'm not sure if it has already, but it will be. They had 18 tanks with breeder pairs, and they rotated them through the grass and the leaf plants and every week in the control. They didn't experience any deaths, but they did have a higher yield of eggs in the tanks with the grass. And they're, well, why? Well, possibly the grasses are more like their natural environment in the Ganges River, where the zebrafish came from. Don't know. But as the zebrafish got older, there was less difference between the leaf and the grasses. Maybe because they just became more seasoned fish, or maybe the novelty of the grass versus the leaves wore off. Also, fish can see colors, so do they have a preference? So, though this test has the presence of the marbles in the tank, the preference for certain colors can be useful in ways outside of the tank. So in this study, they studied clear, red, black, green, yellow, and blue marbles with clear as a control. And the fish really liked the clear, which is over on the left side here. It's very hard to see, but definitely preferred the red, and they didn't like the blue at all. So this group uses this system every day. They put red mats underneath their crossing tanks. And this is especially helpful if they have difficult breeders, such as mutants or older fish. They feel they've had better and faster success, and they've saved several lines this way. And this is an example of how, excellent example of how you use the environmental enrichment outside of the tank so you eliminate your contamination concern. This group is planning to repeat the experiment with EthoVision to obtain more concrete data, which will be good because other people have reported that they found fish react strongly and aggressively to the red and yellow colors. Also, this particular slide is on the difference between the black and the white preference of the adult fish. They found under experimental lab conditions that the fish have a preference for the clear and the black. They had the lower cortisol ratings here. And this was when they housed them at 10 individuals per 2 liter, or 5 individuals, come down to 5 individuals per liter stocking density of water volume, and they were able to express their normal behavior and avoid increased cortisol stress reduction. This is the white here that had the highest stress on the right. And then when you start looking at depth, black, white, and depth, the choice between the black and the white sides of the novel tank, the fish preferred the black side, when the deep and the shallow sides of a novel tank preferred the deep side. The preference when you put them in, they had the weaker preference for the deeper side in the black tanks, he's up here, and the strongest preference for the deeper side in the transparent tanks. Transparent tanks is also dependent on people being in the lab, what's going on around them, and the whatever is on the counter next to them. There's historically been a preference, general preference, to include the use of live feeds, but that's decreasing over the past few years, and that must be weighed against possible health concerns and contamination of your system. Your Artemia feedings have, or thought to, feedings to larvae and adults stimulate the zebrafish to hunt and provide a species-typical behavior. But again, you really have to take in the contamination of your system into account. Also, it's very labor-intensive. The marine rotifers production on the right has been shown very beneficial for early-stage feedings and increased growth rate. definite increased growth rate. One thing there, it's a little bit easier to do some quality control on your rotifers versus your brine shrimp. And of course, with all animals, it's important to maintain stable light cycle. Malfunctions in lighting programs interrupt breeding have definitely shown to cause stress, resulting in an increase in opportunistic disease. Also, you have the even distribution with your wall washing systems, your light washers, so that you have the fish at the top aren't exposed to the real bright light and the bottom have no light at all, and keeping it at a lower intensity. Also, the use of the gradual increase and decrease of light in a dawn to dusk approach eliminates the startle reflex, but also of the lights coming on or off, which may inhibit the breeding. But also, the zebrafish are crepuscular breeders, as Jim was talking about, and those are the animals are those that are active primarily during twilight, the period before dawn and after dusk. So if you mimic your time that you ramp up and you ramp down, that's most reproducing their natural environment. It was interesting when I was putting together this talk, I went back and looked at as far as the publication dates of and the references that I used, and how many of them were over the last 5 years. I didn't find any, well, I didn't use many from before that. So it really illustrates how the behavior of zebrafish is being documented in the last 5 years instead of just, I think they like this, or we've seen this, that they're actually putting into scientific papers. So conclusion, there's more work to be done. There's a lot of work to be done. It's going to require thinking outside the box and having an open mind, then that will be needed. There's a wide variety of enrichment types, and scientific determination and evidence of benefit is needed as opposed to anthropomorphic perceived enrichment. The result will be healthier fish and good science. I'd like to thank the following people for their participation and contribution to this presentation. My references, and we'll answer questions in the breakout sessions. Thank you.